Namaste to all of you. This is Jayvan Chalison from Department of Physiotherapy and Diagnosis from Vidyapeeth University. So, for today's topic, I'll be talking about the absorption of vitamin A. Now, beta carotene, it is cleaved by dioxygenase to form retinol, and the retinol is reduced to retinol by NADH or NADPH dependent retinol reductase, which is present in intestinal mucosa. So, intestine is the major site of absorption. Now, the absorption is along with other fats and it requires bile salts. And in biliary tract obstruction and steatorrhea, vitamin A absorption is reduced. So, within the mucosal cell, retinol is re-esterified with fatty acids, incorporated into chylomicrons, and transported to liver. In the liver cell itself, vitamin A is stored as retinol palmitoid. Now, how is it transported from liver to tissues? So, the vitamin A from liver is transported to peripheral tissues as transretinol by retinol binding protein, that is your RBP. So, one molecule of RBP will bind with one molecule of retinol. So, in the case of vitamin A deficiency, the RBP levels will fall. So, how will it be uptake? So, the retinol and RBP complex, it binds to specific receptors on the retina, skin and gonads and other tissues. So the RBP, it does not enter in the cell, and inside the cytoplasm of the cell, vitamin will bind to cellular retinoic acid binding protein, that is CRBP, and finally to hormone responsive element, that is your HRE of DNA. Now, thus the genes are activated. So, next most important thing in vitamin A is your Wald's visual cycle, and that is Wald visual cycle is, we can also say, the biochemical role of vitamin A. So, first one is generation of nerve impulse. So, Wald was awarded Nobel Prize in 1967 for identifying the role of vitamin A in vision. And rhodopsin, having molecular weight of 35,000 daltons, it plays the pivotal role in vision, and it is a membrane protein found in photoreceptor cells of the retina. So the rhodopsin is made up of protein opsin and 2-cis retinal. So when light falls on the retina, this 2-cis retinal, it will isomerize to all trans retinal, and a single photon will excite the rod cells, and the photon produces immediate conformational change, and the unstable intermediates produced are rhodopsin, which will change to beta-rhodopsin, which will change to lumirhodopsin, which will further change to meta-rhodopsin 1, then to meta-rhodopsin 2, and then to finally opsin plus all trans retinal. So each of these intermediates have a lifespan of only few picoseconds to microseconds. And the all trans retinal is then released from protein. So visual pigments are G protein coupled receptors and 2-cis retinal, which blocks the receptor protein, which is opsin, and in its inactive form. So the isomerization and photoexcitation will lead to activation of G protein and generation of cyclic GMP. Now, cyclic GMP will act as the gate for cation-specific channels. So, transducin in G protein is the G protein in retina, and the nerve impulse thus generated in retina is transmitted to visual centers in the brain, and the signal is terminated by phosphorylation of a serine residue of activated rhodopsin by an enzyme rhodopsin kinase, so that the inhibitory protein, which is beta arrestin, it can bind and inactivate the rhodopsin. So now regeneration of 2-cis retina. So after dissociation, opsin, it will remain in retina, but transretinal, it will enter your blood circulation. Now later, cis retinal is generated, which reaches retina, and the reattachment of 2-cis retinal to opsin is critical for the shutting off pigment's catalytic activity. So the transretinal is isomerized to 2-cis retinal in retina, and itself in the dark by enzyme retinal isomerase. So this reaction is taking place in retinal pigment epithelium, and the two cis retinal can combine with opsin to regenerate rhodopsin. Alternatively, all transretinal is transported to liver and then reduced to all transretinol by alcohol dehydrogenase, which is ADH, and NADH dependent enzyme. So ADH will contain zinc, and therefore zinc is important in retinol metabolism. Now the all trans retinol is isomerized to 2-cis retinol and then oxidized to 2-cis retinol in liver. And this is then transported to retina, which completes the wald's visual cycle. So what is dark adaptation mechanism? So for the work in information processing in visual pathways, Thorsten Wiesel and David Hubel were awarded Nobel Prize in 1981. So rods and cords basically act in dark adaptation mechanism. So bright light, it depletes the store of rhodopsin in rods. Therefore, when a person it shifts suddenly from bright light to dim lit light, there is difficulty in seeing. For example, while you enter the cinema theater, so after a few minutes, rhodopsin is resynthesized and the vision is improved, and this period is called as dark adaptation time. Since this is increased in vitamin A deficiency, red light bleaches rhodopsin to a lesser extent. So doctors used tinted glasses during fluoroscopic X-ray examination of the patients, and rods are for vision in dim light. So in retina, there are two types of photosensitive cells, the rods and the cones. Rods are responsible for perception in dim light, and rhodopsin present in rods is made up of 2-cis retinal plus opsin. And deficiency of cis retinal will lead to increase in dark adaptation time and night blindness. And in humans, one eye contains about 120 million rods, and each of which will carry 120 million molecules of rhodopsin. So this number of rod is more in cats, mice, and owls. Now, what are cones for the color? So cones are responsible for vision in bright light as well as color vision, and they contain the photosensitive protein, which is conopsin. And there are three types of cones. Each is characterized by a different conopsin and is maximally sensitive to either blue, cyanopsin, green, iodopsin, and red, porphyropsin. So in cone proteins also, 2-cis retinal is the chromophore, and reduction in number of cones or cone proteins will lead to color blindness. And in one eye, it contains about 6 million cones. So now color will have profound influence in light. So about 1/3 of gray matter of the brain is involved in processing of visual information, and about 70% of information inputs to the brain are visual. So the optimists view the world through rose-colored eyes, and when the person is in blue mood, saffron color has tranquilizing effect, and especially in agitated persons. So even briefly display of saffron color, it produces measured relaxation in muscles. Now blue color, although makes one peaceful and content increase in muscle tone. So mice living in red light are also most active when green light, they are least active. And other than actual color, the subtle shift is more responsible to unblock automatic impulses, and the effect of colors on human moods have applications in criminology, psychiatry, and interior decoration and chromotherapy and yoga. So this was all about the vitamin A and wald visual cycle. Further about vitamin A, we'll discuss in next section. So until then, if you have any doubts, you can contact me. So until then, take care and goodbye.